topic relating to role of chlorofluorescence in different ABB9 constructs of transgenic maize for drought stress tolerance in the field. Actually, my work is relating on the field. I have conducted different phenotypes, just like chlorofluorescence, NDVI, ASI. So from my PhD work, I'm doing PhD. Almost my PhD is going to be finished from Chinese Academy of Agriculture Sciences with the collaboration of my institute in Pakistan, Sindh Agriculture University, Tanojam, Pakistan. My name is Javed Hussain, and this is my email. This is the outline of my research. Introduction: the worldwide annual cereal production is 2,521 million tons, and globally annual maize production is 1,313 million tons. The drought condition in China, the drought condition of different parts of the China, because I have done research in China, so this is the conditions, very worse condition in China, some coastal areas of the China. Now, actually, the drought mechanism takes places through when there is a low water quantity, and the plants will signaling itself ABA, it's ABA, and the stomata will be closed at that time in stress conditions, and the carbon dioxide will be diminished. So when there's a low water potential in tissues, the rubisco activity will be binding, it will be inhibits, and finally it will be lowered. And with that, also other enzymes are also to be affected. Then a reactive oxygen species also produced in more quantity, and they will attack on the different membranes, just like cytoplasmic membrane or thylakoid membranes, that finally limited the carboxylation, and the process will goes to down regulation of non-cyclic electron transport, and the ATP will disturb, and photosynthesis will be declined. So when there is a high drought, the reactive oxygen species will break down the chlorophyll and green biomass, and finally it will reduce the yield. This is the slide showing the C4 chloroplast. So the C4 chloroplast showing the mesophyll and bundle sheath cells. The bundle sheath cells are inside, very inner part of the cell, and the atmospheric oxygen will not be diffused directly into them. And beside this, the granum and thylakoid membrane showing the different structure of the stroma, which are the sites of photosystem 1 and photosystem 2. Which are responsible to producing the ATP from the stroma and granum. This is the membrane which are ruptures in the drought conditions. This is the by splitting of water into hydrogen ion and oxygen ion, and then electrons will be transferred through the different proteins, proteins are other kinds of proteins depend upon the species, because each species have its own structure, and then hydrogen atom will be pumped inside the vesicles, and flow of electrons takes places, and finally it will produce the ADP or ATP in the presence of inorganic phosphate and hydrogen ion. The mechanism of drought will depend on the signaling of perception and transcription of ABB9. ABB9 transcription control through different transcription factors for gene activation. The gene activation actually depend upon the osmolytes, antioxidants, molecular chaperones, and ion channels. And with these equilibrium state, the gene can perform well, and it will said to be tolerant in adverse conditions. This is the physiological and molecular mechanism for drought tolerance. This is some literature of ABP9, ABR2 binding factor for ABP9 protein using ABR Cat1 promoter as a yeast for hybrid screening of maize, CDNA library. ABP9 encodes a transcription factor of B zip family capable of transactivating expression of downstream reporter gene suggested by Wang. As ABP9 expression improves the photosynthetic capacity of transgenic plants under stress conditions, and it will also enhance in abiotic stress, including drought, salt, and oxidative stresses. The objectives of my study are to find out the transgenic events with higher chlorofluorescence and grain yield among transformed constructs of PABP9, ABP9.1, PUBI, ABP9.1, PUBI, ABP9.2, and PUBI, ABP9 3-flag under drought stress condition at vegetative stage in the field. This is the experimental details, 4-row, 3 treatments in field condition, drought stress applied at V3 and V4 stages. This is the drought was imposed, and the drought was appear from here, and it goes up to 150 centibars. That was the time of male flowering. The transformed different 4 constructs was tested for chlorofluorescence and grain components. The chlorofluorescence measurement was recorded by this method. These are the results. The chlorofluorescence analysis of PABP9, ABP9.1, transgenic maize under drought stress. The lines of the transgenic lines showing a lot of chlorofluorescence and 201, 206, 212, and 214 as compared to wild type. The other transgenic lines also showed higher chlorofluorescence, but these values are non-significantly higher. Then this is the grain parameters, so 100 kernel weight and average year grain yield was noted same from those lines who have performed well under drought conditions at that time. And this is the construct PUBI ABP9.1 showing 611, 612, 616, 617 possessing significantly higher chlorofluorescence than non-transgenic events. Similarly, the 100 kernel weight and average grain yield were also noted in significantly higher in 611, 612, and 617 of PUBI ABP9.1. Then the number 7 shows the construct of PUBI ABP9.2 transgenic under drought stress was assessed for chlorofluorescence of 707, 714, 717. These events shows maximum green biomass or more chlorofluorescence as compared to wild type events. Similarly, these events showed better performance in terms of yield of 707, 714, 717 as compared to non-transgenic controls. This is the number 8 constructs. 809 and 815s are said to be a PUBI ABP9 3-flag leaf was assessed in drought condition in the field condition and showing larger quantity of chlorofluorescence as compared to non-transgenic events. And these same events also showing the maximum grain yield as compared to non-transgenic events. The conclusion of different lines showing 201, 206, 212, and 214 of PABP9, ABP9.1 are said to be tolerant, and similarly PUBI ABP9.1, 611, 612, 617, they showed maximum performance in the field condition in terms of green biomass and lot of chlorophyll. 707, 714, 717 of PUBI 9.2 showed better performance, and 809 and 815 also showed the PUBI 9.3 flag showed better performance as compared to non-transgenic. So it should be said that the research of ABB9 concerning in maize drought tolerance of 4 constructs, PABB9, ABB9.1, PUBI ABB9.1, PUBI ABB9.2, and PUBI 3 flag may be implied for dissection molecular mechanism for maize breeding improvement for drought tolerance for further. Thank you.
It's very quick and short. Thank you.
Thank you, sir.
Any questions? Yes.
Sir.
The different drought tolerance of the different transgenic events that we analyzed, does it simply translate to a higher or lower expression level of the transgene in those lines?
Yeah, higher overexpressed lines were analyzed in the field.
So they are all expressed in all lines. The transgene is expressed
Yeah.
at the same level. Or did you check for the gene expression in these transgenic events?
No, I haven't
Okay.
checked yet.
Right, because it might be just
Yeah.
simply expressed more or less in the different lines. You get much variation within
Yeah.
those lines.